Alrighty, let's start this lecture. So this will be a lecture series of veterinary immunology and avian pathology. Okay, we will be covering veterinary immunology from Ian Tizard. Actually, the notes which I have prepared from Ian Tizard, these are the notes of first chapter which I have prepared. Every day I will be uploading two lectures, one of immunology and one of avian pathology. So hope this helps you. So veterinary immunology from Ian Tizard. Let's start. So before starting, I want to show my Ian Tizard. So this is my Ian Tizard. It is the 5th edition and Indian edition actually. So we'll discuss from it later. But first, let us start with the basics. Okay. So the defense of the body. So first we will describe what is immune system. How will you define immune system? Immunology, as we know, is the study of immune system. Immuno means immune and logy means study. The study of immune system is called immunology. But what is actually immune system? So immune system is a basic system which is found in the body of animals, which helps in the protection of body against foreign microbes or foreign pathogens. Okay. Now one of the most important term in vaccination or in immunology is vaccination. We will discuss this point later, but we will move on to this point. One of the most important point in immunology is vaccination. One of the most important development will lead to a further development of immunology is vaccination. So before discussing the principle or definition of vaccination, we should know what is VECA, from where the term VECA has derived. Vaccination has been derived. So vaccination derived from the term VECA, which is Latin for cow. Okay. So what is the principle or definition of vaccination? So here in vaccination, what we do, we expose an animal to a strain of a particular microbe that will not cause disease. We know that will not cause disease. We have either attenuated it or we have reduced its pathogenicity so that it does not cause any disease in the animal, but it can provoke an immune response. So this is the main reason that why, what is the principle behind the vaccination. So what we will do in vaccination is we will expose an animal to a particular strain of a microbe which will not cause disease, as we know that, and but that will provoke an immune response in the animal. So that will help in vaccine. That is the principle or definition of vaccination. Now this immune response will protect the animals against subsequent infection or by the same or closely related strains. Okay. Now let's move ahead and let's check this point. So that immune response will be of low intensity, and then the natural infection and the inoculated animal will become resistant to that disease. So what this point basically means is that so this point means that when we will expose an animal to a particular strain of microbe that will not cause disease, so that will lead to what? That will lead to the development of a natural resistance, or not natural resistance. That will lead to a development of an immune system, immune response. That will be of low intensity, but then the natural infection, that intensity will be less than the natural infection. That intensity due to vaccination will be less than the natural infection, and this all will lead to the animal become resistant to the disease. Okay. Now there are four important names: Louis Pasteur, Salmon, von Behring, and Kita-Sato. Okay. So Louis Pasteur, he made the key discoveries that led to the development of vaccines against infectious agents. Salmon demonstrated that dead organisms can also be used as vaccines. Von Behring and Kita-Sato showed that bacterial products like toxins will also be used. So what these 3 points basically means is that Louis Pasteur was the person who actually made the key discoveries which led to the development of vaccines against infectious agents. So this can be asked as an MCQ. Next is Salmon. Salmon demonstrated that the dead organisms can also be used as vaccines. And von Behring and Kita-Sato, they showed that bacterial products like toxins could also be used for vaccination purposes. Okay. Now the body's defenses. So there are 3 lines of defense: first line, second line, third line. So first line is physical barrier, that is skin and mucous membrane. Second line of defense is the secretions of the body, which I haven't written here, by the way. So secretions of the body, for example, the acids in the stomach, the bases in the small intestine, basic pH of small intestine, the saliva, the tears, all of this. And third is the cells. Third line of defense is the cells. So cell consists of this inflammatory cells, that is neutrophil and monocytes, and which perform 2 functions. They destroy the invading microbes and they prevent the spread to the uninfected body parts. Obviously simple. These cells, inflammatory cells, or the third line of defense, will destroy the invading microbes and they will prevent their spread to the uninfected body parts of the body. Okay, now this is a simple diagram just to represent the role of every line of defense. So these are the invading microbes represented by 3 arrows, that is large in number. These are very large in number. Physical barrier removed some amount of microbes. Then for second line of defense, that is secretions of the body, removes further some type of microbe. And the specific immunity, that is the cellular line of defense, removed all of the microbes from entering the body or destroyed all of them. Okay. Now, next point is the immune system can recognize the foreign invaders, destroy them, and retain the memory of the infection. Just like we know that in physiology, veterinary physiology, we have learned that the neuron has 3 functions to perform. Okay, what are the 3 functions of neuron? Detect, transmit, and detect, receive, and transmit the impulse. Here, in the same way, immune system has 3 functions, that is to recognize the foreign invaders, to destroy them, and to retain the memory of the infection. Okay. Now these we know: bacteria, most bacteria grow outside the living cell. Virus can grow only within the living cell. So these are basic points. Now invaders can be divided into 2 categories: endogenous and exogenous invaders. Endogenous, as the name suggests, which originate from inside the body of animal, and exogenous, which originate from outside the body of animal. Endogenous example are cancer cells, and exogenous examples are bacteria, protozoa, helminths, parasites, etc. Helminths means parasites. Okay. Now types of immune system. Okay, so types of immune system, there are 2 types of immune responses: humoral immune response and cell-mediated immune response. So what does it mean of humoral here? Humoral means any body fluid. So this takes place in the blood, so that is why it is called humoral immune response, which leads to the production of antibodies, and they destroy the exogenous invaders. On the other hand, cell-mediated immune response destroy the intracellular or endogenous invaders, and use of cytotoxic cells is there, for example B cell and T cell. Okay, so this is antibody mediated cell immune response, and this is cell mediated immune response. First we will discuss the antibody mediated immune response in detail. So the protective factors found in the serum of an immunized animal are okay. So this point means that these 2 points. These 2 points we need to understand, these 2 points, because these 2 points discuss the very basic terms, that is antibody and antigen. So antibody, the protective factors found in the serum of an immunized animal, which are protein in nature, are called antibodies. Okay, and antigen, the substances which stimulate the antibody production, are called antigens. The substances which stimulate the antibody production are called antigens, and the substances which are protein in nature and are formed after by the stimulation of antigen are called antibodies. Okay. Okay. Now the antibodies can only bind to, okay, this point is not of importance. Okay, what is a toxoid? Chemically detoxified toxin. Okay, now this is a graph. This graph is important. So this arrow means the first dose of antigen, this arrow means the second dose of antigen. Here it is written the titer, that is the amount of antibodies in the serum. So this in itself is a question: what is titer? The amount of antibodies in serum. Now primary immune response, secondary immune response. So primary immune response, it means that primary immune response is slow and is of low intensity. On the other hand, secondary immune response is of high intensity and lasts longer. Okay, so this all is written here. So what are the features of secondary immune response? What are the features of secondary immune response which we have seen here from this graph? So the features of secondary immune response are as follows: they occur more quickly, antibodies reach very high level, response lasts much longer, and can be provoked by a second dose of antigen only. Okay. Okay. Now, also secondary immune responses are also called anamnestic responses, because anamnestic means having memory. So secondary immune responses will have memory, so that they are able to recognize any foreign pathogen when it enters the body. Okay. But this point is also important: repeated injections of antigen do not lead to indefinitely to greater and greater immune response. So this does not mean that it will again and again inject the antigens and that will lead to greater and greater immune responses. This is not the case. Okay, so this was antibody mediated immune response. Next is cell mediated immune response. So here there are 2 set of reactions. So what we discuss is antibody mediated immune response. We discussed in antibody mediated immune response that there are primary immune response, secondary immune response. The secondary immune response occurs more quickly, antibody reach very high levels, response lasts much longer, and can be provoked by second dose of antigen. But in cell mediated immune response, what we will discuss is, here there is 2 set of reaction, that is first set reaction and second set reaction. Okay, so we'll discuss the example. So suppose we have 2 dogs of the same breed. So first is a donor dog, second is a recipient dog. Okay, and we transplant a piece of skin into the recipient dog. Okay, and then that piece of skin will survive for 10 days in the recipient dog. Okay. Okay, so where we were here that we are studying cell mediated immune response. Here there are two set of reaction. First set reaction, second set reaction, and we need an example to explain both of these. So example is as follows. Suppose you have two dogs of the same breeds. So one is the donor dog and another is the recipient dog. So what we did, we took a piece of skin from the donor dog and we transplanted that into the recipient dog. So that piece of skin or graft, as we call it in immunological or medical terms, so that graft will survive in the recipient dog for only 10 days. So this response, this first set of reactions, so after that will be rejected, that graft will be rejected. So this set of reaction, this first set of reaction, which is analogous to a primary antibody response, because it is slow, it is a weak process, this is called first set reaction. Okay, now if we again take a piece of skin from that same donor dog and transplant it into the same recipient dog, then the second graft will not survive for more than 1 to 2 days. Why? Because there is a second set of reaction. It will be rejected after 1 to 2 days because of second set reaction, which is analogous to secondary immune response, which is fast and powerful. Okay, now these points are also important because as we know that for immunization or vaccination purpose, we can use either live organism, attenuated organism, or live attenuated organism, or dead organism, any part of organism, any virulence factor or toxin produced by organism. But for graft transplant, we can only use the living cells. Very basic point. Okay. Example of what is responsible for graft rejection? The cell mediated immunity. This is very important. Okay. Next, you have the control of immune system. So immune system is tolerant for the normal tissues. The immune system of animal or human, any vertebrate, is tolerant for normal tissue. Tolerant means that it can tolerate its own tissues. It can differentiate between self and non-self. Okay, there are two processes involved: negative selection and positive selection. Negative selection, here those immune cells can respond to self antigen that they are destroyed. Meaning, in negative selection, that is, those cells are destroyed here which can respond to self antigens. Okay, so as we know that each of the body cells also has some antigens. For example, RBC antigen present on the surface of RBC. So here those immune cells that can respond to self antigens are destroyed, so that they could not respond to self antigens, and thus they cannot destroy the self cells. Okay, body cells. And positive selection means that here the surviving cell that can recognize the foreign cells are allowed to grow. Okay, the foreign cells that are allowed to grow. Okay. Now, mechanism of immune responses. So there are two functions of immune responses, two mechanisms: humoral response and cell-mediated response. Okay. Okay. So the important point here is this. This is not at all important. This is not at all important. So humoral response is responsible for keeping foreign cells out, and cell-mediated response is responsible for keeping internal balance. Okay. So this was the first chapter from my notes, which were made from Ian Tizard, and let us discuss this from Ian Tizard itself. So this is my Ian Tizard 5th edition. So let us open it, the first chapter, and here's the first chapter. So we have already discussed these underlined points. Okay, so these underlined points are discussed. See, this is an example to show that what does this example show? That Louis Pasteur, what he took some strains of Pasteurella multocida. Okay, and those strains are old, aged strains. And so then he injected those strains into some chickens, normal chickens. So that chicken survived. Okay, so this was a kind of surprise for Louis Pasteur, because it was known that Pasteurella multocida kills normal chickens, but there was no death or disease. Now what he took was, he took these same chickens because he was short on funds, and then he took again fresh sample of Pasteurella multocida and injected into the same chick. That also survived. But when he took fresh Pasteurella multocida sample and injected it into a normal chick, another chick, then it was dead. So in this way it shows that yes, immunity has memory, and immune is also called an anamnestic response, secondary responses. This diagram, as I have already discussed from my notes, this is the diagram. So microorganisms invading microbes, physical barriers, stopped some of them. Focus responses, second line of defense again stopped some of them, and then third line of defense involved stopped all of them. Okay, so next is this diagram, this graph right here. As we already discussed, this is a first antigen injection, this is a second antigen injection. Amount of titer, serum into antibody in the serum, that is titer. Primary immune response, this is a lag period. This is primary immune response, weak and of low intensity. Secondary immune response, high, very strong and of high intensity. Okay. Now this is the characteristic time of course rejection of foreign skin graft, which is similar to figure this figure. These two figures are very similar. These two figures are very similar. Okay, this and this figure are very similar. Okay, so all of this we have discussed. Thanks a lot.